Hello, this is Sylvain Coste at UC Berkeley Faculty Club, and today we are meeting with Swati Girdani and Rainer Sachs, and we will talk about a review they wrote recently for radiation research. Well, welcome for a very special podcast. It's actually a video cast today, and I am with Swati Girdani and Rainer Sachs at UC Berkeley at the Faculty Club. And we're here today to discuss the recent review they wrote entitled Biological Effects of Protons Radiation, What We Know and Don't Know. So thank you for being here.
Thank you for having us.
So Ray and Swati, why don't you tell us a bit about why you wrote this review? Can you give us some feedback?
So, one of the projects like we are working with Lynn Halatki at Tufts University is on the effects that proton radiation have on various hallmarks of cancer. And so we started this work a few years back, and one of the amazing things that came out was that proton radiation in the initial gene microarrays that we did was that proton radiation was downregulating a lot of proangiogenic genes, and which was kind of like surprising because the conventional belief of with gamma radiation is that a lot of these genes are upregulated after radiation, after the conventional gamma radiation. So that started a whole process of thinking that the entire field believes that proton and gamma radiation are exactly similar, even though they have different dose deposition characteristics. I mean, one is a particle radiation, one is not, so one is a particle radiation and one is not, and because the cell killing and survival and clonogenic results are very close to each other, so it was taken for granted that the entire biology is very similar between these two radiation types. And when we started seeing results in our own lab that they were behaving very different from each other, that became the motivation about going back into the literature and looking for if somebody has found something similar, and compiling all of them together at one place.
So overall, what's the conclusion? We're going to go in details, but do you think that there's overwhelming evidence that says that they are very similar in terms of action, or in fact that there is a lot of evidence going against that? What do you think if you were to judge that? Is that a 50-50, or it's more like 80% of the endpoints measured suggest it's the same? Or why is this concept out there that it's equal, very close?
Well, of course, it's only fair to restrict attention to low LET protons when making the comparisons with X or gamma rays. There, Swati will know better, but there it looks like more like 50-50 as far as the data
I would
is
take
concerned.
a more cautious approach and say that there hasn't been that much of research done, and whatever is out there, it's definitely warrants more work. It definitely begs us to not get fixed in a dogma, and to think a little outside that they could be different. There's enough differences reported for us to keep on exploring. That I'll take a more cautious approach to it, I guess.
You want to add something, Ray?
No, I mean she's certainly right on that. Yeah, we don't know enough yet experimentally.
I mean, because they haven't been even clinically, they haven't been randomized clinical trials done together, and the availability of proton beams wasn't all that much till now for research purposes, and it's now at Brookhaven, but the whole feasibility of trying to do experiments at the same time under various controlled conditions has come up that has become, I think, more possible recently than it was at the time. So now you can really plan those experiments and try to answer those questions better.
So I'm going to ask a very simple question. In terms of comparing protons and gamma rays, and I understand low LET protons, when I say protons I mean low LET protons, what are the endpoints that seems to show very little difference between low LET protons and gamma rays or X-rays? And what are the endpoints that you've seen in the literature where differences are extremely large? And you can talk about your own research if you want there as well.
I think with respect to survival and clonogenic survival, so with respect to cell killing, that's where the maximum amount of research exists, and that's where there hasn't been that much of differences reported between the two radiation types. When you take low LET protons and like similar LET protons and gamma into consideration, there's been a lot of work done on it, and mostly people are of the consensus that the RBE of protons is around 1.1 to 1.2 in the same range, so it's not drastically different from gamma. That I think everybody accepts in the field because of the amount of data that is there. I think with respect to other aspects, again, I'll take a more cautious approach. There haven't been that well studied. I think the new aspects of gene expression analysis, they have been finding various differences between proton and gamma, and that is something that is a little difficult to ignore, because gene expression really tells you something that the biology could be really different. With the rest of the other points, I think there's some literature out there mostly saying that there could be differences, and we should not take for granted that the survival or the RBE with respect to cell killing being so similar should be taken for granted, with the biology being exactly same for both of them. So that would be.
Ray, do you want to add something?
Well, yeah, I mean, I think maybe Swati will correct me, but it's mainly tissue level responses, including angiogenic responses, which fail to indicate similarity, if I put it as cautiously as.
I mean, that is our research. I don't want to advertise our research. It's I'm just talking more broadly about the field. There are probably only a couple of papers with respect to angiogenesis and invasion, comparing gamma and proton, and they have found differences. But
would you mind summarizing at least your results, since you know them pretty well? Those are your results. Granted that that's not.
Again, with my results, I would just say that we did one part of the proton results at BNL. With the low LET protons, and the gamma work was done back there at Tufts University. So they were done at two different time points, and you can take them, but they were not controlled experiments done at the same place. And we have found differences with respect to gene expression. We found that proton downregulated a lot of important pro-angiogenic, pro-invasion genes like VEGF, HIF1 alpha, IL-8, IL-6. We studied cell invasion after proton radiation and found that proton radiation downregulated cell invasion, both directly hit cells as well as in a bystander kind of a setting. We've irradiated mice with proton radiation, taken out tissues, and then even done molecular staining for and RT-PCR for a lot of these factors earlier reported, VEGF, HIF1 alpha, IL-8, and again found the differences. And there's enough to say that there were, and we've done similar experiments with gamma and found the opposite results. But yes, we haven't plated the cells at the same time, irradiated them kind of close to each other with protons and gamma at the same location. So that's one thing that I don't believe that would make a difference, but again, it's not a controlled experiment in one setting.
Well, there's a simple way to deal with this. Is could you repeat some of those experiments at Brookhaven with the gamma source they have there, and
that's what,
and confirm that the gene you picked up in the first time are still there at Brookhaven?
Yes, that's what we plan on doing in the coming runs. And one of the things which is really great, lately they've come up with a new photon source where you can control the dose rate. So now we're going to even have exactly similar dose rates. So we're going to see what we're going to get for protons and use the exactly similar dose rates for the photon radiation too.
Right, so I heard about that. So Ray, since you're the modeling expert in this field, would you maybe speculate or tell us what you think is happening, why the tissue response is different, and why angiogenesis? Do you have any idea of a mechanism behind this that explains that difference?
Well, of course, I was interested really in the discrepancy that those results show compared to the standard micro and nanodosimetric arguments that low LAT protons are essentially the same as the low LAT electrons produced by gamma rays, and therefore you're not allowed to have any differences on an extreme, I mean a theoretical microdosimetric view. That's just not allowed. And I sort of thought, well, are there any differences between the radiations? And of course there are. Yeah, the protons travel mainly in straight lines at very low doses. There's a very different kind of dose localization for the protons on a millimeter scale. Yeah, so that the similarities which people have heavily emphasized on the micrometer and nanometer scale, and I think perhaps implicitly taken for granted that the targets, the basic targets, are on that scale, non-interacting targets on that scale, that the known differences between the radiations might be coming into play, and therefore one should look at the experiments with unprejudiced, I mean, without paying too much attention to the theory itself. Now, the differences are that in a tissue might, for example, respond to the directionality of a proton beam from one angle, whereas for photon irradiation, that's not a plausible possibility. And that at very low doses, as I said, there's much more localization for the proton beam along a single line. And for very large scales, you even get situations where let's say you have a situation where one proton hits a tissue scale, an interacting tissue scale organ, or which is heavily in communication with itself, and all bits of which influence all other bits. Then at a very low dose, you can have a situation where a single proton hits that during one relaxation time, and then the dosimetry is really very different. So I guess my conclusion was you have to look at the experiments without assuming a priori that the standard microdosimetric arguments are necessarily valid.
So you mentioned a single track. So I'm thinking one difference that would come to mind between gamma rays and X-rays and low LET protons would be that track structure. And so something that comes to mind is the bystander effect, where you have one series of cells being traversed, then another series of cells not being traversed. With gamma rays, no matter how low you go, I mean, unless you go extremely low, you can assume a fairly uniform distribution. So I'm wondering, do you think bystander effects are involved in those differences?
Yeah, it could easily be that well, I mean, one track could hardly do it in the relevant time scales for bystander effects. But if the first track comes along and the other track comes along, the next track comes along a measurable time later, then the fact that the first track was in a straight line through this now huge, I mean, it's a very narrow straight line through a very large volume, as opposed to electron energy deposition scattered throughout the volume randomly. Yeah, if the first track wakes up the tissue, yes, then the bystander effects could be, in my opinion, markedly affected. Yeah, and I think a tissue can communicate with itself very rapidly. Yeah, and you don't always have to wait for diffusion. There are it's known there are electromagnetic communications between cells in a tissue.
Yeah, but one of the, when you in your review, Figure 5 discussed the old paradigm and the new paradigm, for instance, and so that's focusing on the new paradigm. I think the concept of angiogenesis being inhibited by the protons. Is that correct?
Yes.
So, which is a positive effect as far as the system is concerned, right? Because you're blocking angiogenesis. Angiogenesis is not something you want to have to happen. Do you agree with this?
I mean, with respect to tumor angiogenesis and even with carcinogenesis risk for NASA, it's definitely positive.
So, and that's really where, and again, maybe I'm putting things in too many boxes and we need to think outside the box, I understand. But the classic box for bystander would be, bystander is the amplification of something bad. Adaptive response would be the opposite, right? Where you have some type of response that protects. So this inhibiting angiogenesis seems to be like something that an adaptation maybe of the system as well. How do you see this? I mean, do you
I don't think bystander is like an amplification of something bad. Bystander is like you as a stander. It could be positive. It could be negative. It's just like whatever is happening close to you, how does that affect you? That's basically what bystander effect is. Let's say the way the cells interact with the proton radiation, they're releasing or they're not releasing a lot of cytokines. I mean, one way of looking at bystander, as I look as as a biologist, is release a lot of cytokine factors, and some of them could be pro-angiogenic and or anti-angiogenic. So how as a bystander you're affected? I mean, that's bystander is not necessarily an amplification of a negative. That's the first thing I would say. It's just you responding to something that's happening in your vicinity.
I see. But so, do you have any idea of what triggers this anti-angiogenesis?
Well, let me go back a minute. I guess I think the view now is that the bystander effect in radiobiology is just sort of a somewhat belated recognition of the fact that cells are constantly interacting with each other by all sorts of mechanisms and on all sorts of scales. Okay, yeah. So I don't. Yeah, I mean, I would say it's intercellular interactions, which are the key mechanism which you want to be talking about, and not necessarily the classical or the now classical radiobiological bystander effect. Yeah.
That's why I was trying to say that I don't want to put things in boxes because those boxes tend to have a connotation, and my feeling is adaptive response is another aspect of that communication.
Right.
But for some reason they've been tagged with the positive and negative aspect in our field, I think, for the majority of things. But you're right. What we're discussing here is something a lot more organic. Is that communication? So focusing on that question of communication, I'm still puzzled by this anti-angiogenesis response and what would be the actual mechanism behind it, because the tissue, the doses you were looking at what range of doses did you look at?
From 0.5 gray to 2 gray.
So fairly high doses.
And Peter Grabham's group from Columbia, he's also looked at angiogenesis in a totally different model than what we have. He's been looking at 3D structures of Hewitt cells and the tube formation in them, and one of the things that he reported was that developing vessel formation was inhibited by proton radiation, but even at doses at lower than 0.4 gray to 0.08 gray, you could see that kind of an inhibition taking place with proton radiation. but when it came to gamma radiation, you had to go to like really high doses to see some kind of an effect in the lumen formation, in the lumen diameters and the tube formation. So it's been reported. I mean, invasion, which is another aspect related to angiogenesis, has been reported by the Japanese group with proton radiation, that it inhibits invasion. It has been like on a different note with carbon ion radiation. There are a number of papers out there which says that carbon ion radiation, I mean, that kind of like takes it to a different aspect, because it's
very different.
It's like, charge is different, LET is different, and you could just say that it's because of due to those effects.
Yes, we're not low LET anymore.
Yeah, so but like, I mean, even for protons, there are a couple of papers, at least, like, the Graham's paper I could really cite, that was excellent work, and the Ogata paper from Japan that showed the same effects. They're seeing a lot of molecular players coming up, like, from a lot of angiogenic factors and MMP9, and I think Graham, Peter, when I talked to him, he was talking about PK, like, protein kinase C signaling. We think it's a lot of VEGF and IL-8 signaling that's coming into play. So a lot of differences in the protein interactions.
So what do you think that all the work the review you've done, and the comparison between proton and gamma rays, do you think we need to revisit certain aspect of so there's two questions, right? There's the risk assessment for the NASA space program, and there's also the radiotherapy, right, with proton therapy. So could you comment on the implication of what you have reviewed for those two fields, which are very distinct?
I mean, for both, for NASA, it is obviously carcinogenesis risk, and the way we look at it, like, the Halatki group at Tufts is like, the astronauts who are going up in space. One thing is very important is initiation of cancer that is taking place. The other thing equally important is the promotion and progression, because as we all know, like, we all harbor a lot of dormant tumors in ourselves. And so, like, the middle-aged astronauts going up there in space, they would have a number of dormant tumors. So how proton radiation and like all these dormant tumors, before they can become fully malignant, clinically relevant tumors or cancers, they have to pass through a number of bottlenecks, and one of the most crucial bottleneck is the bottleneck of angiogenesis, is the bottleneck of cells being able to kind of like, free themselves of the matrix and kind of like invade and metasize, like, go away and like, travel and like, go to other place and colonize. So for that one, for that reason, like, all these hallmarks of progression, like invasion and angiogenesis, are crucial for even NASA's carcinogenesis risk, because as I said, like, they are very important for tumor progression. And the same is when it comes to clinical.
Well, before you go there, so I just want to make sure people listening understand. So having less angiogenesis
is
from
good.
proton is a good thing.
Is a good thing for
it reduces the risk from a space mission as far as that specific endpoint.
I would, yeah, of course.
Okay.
That would be.
And now for radiotherapy, how does that translate?
I mean, radiotherapy, like, there are, like, the reason protons are being advocated in radiotherapy is because they have a superior dose deposition characteristics.
Mm-hmm.
That alone, like, has made them very popular. I mean, there are obviously, like, cost effects that are being debated on whether they are, whether the high cost kind of like rationalize the use of protons, but having the biology in there, like, the protons even having a superior biology, that you are not affecting tumor recurrence, let's say, or even the incidence of secondary cancers later on, especially in case of pediatric tumors where they have a much longer lifespan and higher incidence of recurrence or secondary tumors. If proton radiation affects the biology in such a way that angiogenesis goes down, invasion goes down, that's a definite advantage.
Mm-hmm. Sounds good. Ray, I wanted to also ask you on the NASA project related to this finding. Basically, most astronauts are going to be 40 years old, 30 years old, probably maybe more. So would it be fair to say that the biggest concern is progression and not initiation for those people? Or do you believe there's also initiation is another risk that we should always consider no matter what? The reason I bring this up is because this angiogenesis seems to really focus on something that's already there, that can be just enhanced, which is the dormant tumor idea, right? Which I believe in. But what about initiation? Is it fair to say that those are risk consideration that really applies to a younger population?
Well, of course, yeah. I mean, the possibility of accelerating progression becomes relatively more important as the population ages. You know, there are a lot of factors like what is the latency time for this particular tumor? Like, for example, with leukemias, and then initiation is still a consideration even at quite old ages, because many of them come up very fast compared to the solid tumors. Yeah, for the solid tumors and for ones which have a long latency time, then definitely the progression. I mean, it's a relatively neglected aspect which may turn out to be the tail that wags the dog, yeah, and we have to really look into that and look at the data. The data, even in the case of leukemias, the data can tell you quite a bit about whether you're looking at progression or at initiation, yeah. For example, from the timing compared to the comparing age-induced cancers with radiation-induced cancers, yeah, then the differences in timing give you a clue as to which one you're looking at.
So, I mean, I would just like to add to that when you talked about how this is relevant to NASA, I was basically, yes, focusing on my research, which is angiogenesis, but obviously, like, you know, studying the differences between proton and gamma biologically has its, like, you know, it's even very important for initiation when it comes to NASA's risk. I mean, what we've reported in this review article, that there are differences in like, you know, differences between protons and gamma or photons with respect to DNA damage. There are differences with respect to even cell cycle, apoptosis, different signaling. I mean, we're trying to say that they are biologically different. So obviously, we concentrate more on angiogenesis because that's the thrust of our research.
Of course, yeah.
But like, you know, I mean, all these factors like DNA damage and cell cycle response and gene expression response, they govern a lot of aspects of initiation too. So you don't know. I mean, I mean, yeah, I'm certainly not an expert at, like, you know, the initiation part on how these would impact initiation, like, you know, proton versus the terrestrial radiation. But it definitely opens the field for other people who concentrate on these factors to, like, you know, not just concentrate on not to just get set up in this dogma that it's going to be similar between these two radiation types.
Right. You know, the other thing that it brings up, I think, is when you have antagonistic effect, or, you know, inhibition versus activation, one wonder what happens if you mix those two together, right? Because in fact, back to the NASA mission, they're not going to be exposed only to protons. There's going to be
Yes.
many different exposures. So, did you start looking into this? I know, I'm sorry, I'm taking you a bit out of the review context and more into your work, but I feel it's still a relevant question. Is like, you know, not only proton versus gamma, but what about protons and gamma together, or protons and high LET and so on?
I mean, I thought about that and did a few calculations, and I decided that just simple superposition of effects is at the breakage level, yeah, is such a plausible assumption, and tells you so much, because well, once you know what the breakage is, you think you know a lot of what the consequences are, yeah. So that I mean, it's not for me to second guess what the experts are doing, but my inclination would be to make that assumption and focus on understanding specific radiations in depth, and just live with the fact that I'm not absolutely sure that if I have small dose of gammas and small dose of iron, then I can, by plausible theoretical calculations based mainly on breakage results, I can infer what the mixture should do. So let me say it again. Of course you don't know, but since they're all possible mixtures of all possible radiations, you're also not going to learn experimentally what the actual situation is. You have a fairly simple hypothesis of superposition of breaks, okay? And I would be inclined to use that as a working hypothesis and emphasize the in-depth understanding of individual radiations. But that is a minority view, clearly.
Well, I think it's actually the classic approach. But I'm wondering, you back to DNA break when you think about this, and when we're trying to explain why the protons were different, one of the things you infer was the first track coming in, inducing something, and then sequentially things have changed. So in that context, then it would be hard to approach the problem that way with a mixed beam, because if indeed a track does something different, then into some kind of synergism. It seems that this approach can't work for those tissue response. You know what I mean? It's like I
agree with that. Yeah, I mean, when you ask about superposition of radiations, I guess the first thing is, suppose we try to understand superposition of radiations on the classic DNA-focused level, and then later maybe put in tissue stuff. So yes, my comment certainly was meant leaving aside things about tissues. Yeah, what would you think? Yeah, and the best I could do is to say that this is a comparatively minor assumption that you can deal with two of them if you know how to deal with each one, and therefore in view of the virtual impossibility of looking at all the combinations, or even deciding which are representative combinations or something, I would hope that the emphasis is on individual radiations, admitting that sort of contradicts my comments about tissues being important. Yeah.
So anything you want to add?
I think just conclusion that the whole dogma, what figure 5 represents, that the paradigm is they are totally similar, and just to encourage the field to look beyond that, to when they are doing the experiments, to see that there could be differences between proton and gamma, and just go with it, kinds. I mean, that would be the conclusion part of this, and certainly open up this field for more research.
I think, this is just, Swati knows the answer to this, but my impression was that she set out to do a fairly impartial, very broad survey, yeah, and let the chips fall where they may, but the point was to include everything which met certain criteria, whatever the results. I don't know.
Yeah.
Is that what you wanted? And do you think
I think basically because there's so much of literature out there where people have worked with protons which are low energy, and then compared them with gamma, and just to kind of go through all that literature and really zero it on the literature where they've worked on low LET protons, and try to even do same experiments, and try to compare low gamma also in the same experimental setup. That was the main focus, on zeroing on those papers and then just reporting the results from those papers. Some people have reported no differences, and that has been, I think, impartially reflected, that these people found this, or some researchers found this, and I think we've tried to not bias it to any way, and kept it pretty impartial, just to open up this whole debate.
Well, thank you very much.
Yeah, thank you. So bad.
And